Hello, today we're going to talk about the adaptive immune response. That's kind of a continuation of previously when we were talking about the innate response. We're also going to talk about some things that may happen when the immune response goes wrong, such as what we call hypersensitivities, or which can be associated with like autoimmune disorders, that kind of thing. So let's review our cells. So the innate response, remember the innate response was the non-specific response, but we saw those neutrophils. This is just kind of a reminder. Those were our first responders that come out in kind of high numbers, don't live very long. Macrophages, and these are in purple for a reason. Okay, so macrophages and dendritic cells, if you recall, were cells that could phagocytose or they could eat things. Okay, so they would see foreign debris in there and then they would just eat it. Or engulf it. And then in this case, what we're going to see is that these macrophages and dendritic cells, that's why they're in purple, will actually take the products of what they ate and show it to the T cells up in the lymph nodes. So they're going to move up into the lymph nodes and they're going to work with the lymphocytes. So even though they're technically not part of the adaptive response, they are kind of a messenger to tell the adaptive cells what might be going on. Okay, so the adaptive cells are the lymphocytes, which are B and T cells, and we're going to actually find out that there are two different types of T cells. But they're called lymphocytes because they're found in the lymph nodes. Okay, so even though they're made, so all of the blood cells are made in the bone marrow, but then these B and T cells will eventually kind of migrate and move their way into the lymph nodes and into the spleen, and but they're associated with that lymph system. Okay, and again, the macrophages and dendritic cells, they function as what we call antigen presenting cells. Okay. Okay, so we're gonna figure out what that means, and then they're going to activate those T cells that I mentioned. So even though they're not lymphocytes, they do work with the lymphocytes for that adaptive response. All right, so let's start looking at, I guess, the difference ultimately between the innate and adaptive, but we're going to do just a very quick review here of the innate system. So we know we have to clean up after pathogens. If something comes in, we discussed this when we talked about inflammation. If something breaks the skin, then there's possibly foreign debris that's going to be there. Okay, so there are two basic immune system responses to deal with anytime something invades or moves into our body. That first one is the innate, and it's not specific, so it's going to attack anything non-self. Okay, so when we're going to learn more about specificity a lot today, but in general, the innate immune system looks for patterns. Okay, so these molecular patterns, so pathogen-associated molecular patterns. What that basically means is if, like, it would be like if you saw something that had a tail, you would say it's not human, or if it had fur, it's not human. So that's not really specific. It's not saying, oh, you're the one that committed this crime. It's just saying, well, you're not a person, so you need to go. So that makes it faster. The cells that comprise the innate immune response are anything except these B cells and T helper and T cells. So there are two different types of T cells. Again, T helper cells, CD4, and cytotoxic T cells, CD8. I've written that a bunch of times so we can keep those straight, CD4 versus CD8 and what kind they are. And then also natural killer cells, which are still part of the innate system. So the innate system is, again, anything except these lymphocytes here. T cells and B cells. Okay. All right. We already mentioned doesn't distinguish between microbes, recognizes those patterns, not specific protein sequences, which is how we're going to see that the adaptive immune system does it. Okay. And of course, we covered that when we covered the inflammatory response. So adaptive, also called acquire. So it recognizes and kills specific pathogens. So the way it does that is it recognizes specific protein sequences associated with a specific pathogen. We're going to call those antigens. We're going to look at that. Because it is specific, it can really crank it up. So you don't want to crank up your macrophages just in general and have them just going crazy. But if you have that specificity and you're pretty confident that it's chasing the right thing, then you can turn it up and you can have that stronger, more prolonged response to a specific pathogen. They also have memory. So memory in this case means that any subsequent exposure. So if you get I don't know, a certain cold virus, then you should have. That's probably not a great example because the memory doesn't last very long on those, and they change a lot. But if you get infected by something, then the cells that fight off that infection are going to kind of lay low for a while because they know it's out there, and they're going to come back, and they can possibly come back if that virus comes back or whatever the pathogen happened to be. Okay, so the strategies. So this is how this is going to be broken down. Adaptive immune system strategies. There's a humoral strategy which can kill specific free-floating pathogens. So when we say humoral, we're talking about back in the olden days where they talked about the 4 humors, which were the fluid parts of the blood or fluid parts of the body, the blood, the bile, the other stuff. We don't look at it that way anymore, but that term humor still refers to the fluid portion of the body. So it can kill, the humoral response can kill specific free-floating pathogens, which means pathogens that aren't hiding inside cells. Okay, that's the humoral response. And that involves many cell types, including those macrophages, APCs, the antigen-presenting cells, and CD4 helper T cells. Okay. But ultimately, the thing that distinguishes the humoral response from cell-mediated is that B cells get involved and they will produce antibodies. Okay, so we're going to look at those. Cell-mediated is the other attempt, and those can kill specific pathogens that are inside cells. So that's the difference. Cell-mediated means that the pathogen is hiding in the cell. Humoral means that it's free-floating. Either way, the immune system is going to find them. Okay, so how does specificity happen? How are these T cells and B cells able to say, oh, I'm going to attack this, and how do they know it's not one of your own proteins? Okay, so that's what it's looking at. It's mainly looking at proteins, peptides, things like that. How does it know that? Why don't we just attack and kill our own proteins? That's the question. So again, all blood cells are made in the bone marrow, but a certain type of training happens for B and T cells. So it involves this process called negative selection, which ensures that the receptors on these B and T cells won't recognize and attack the proteins of their own body. They won't attack self. That's the way we say that. So distinguish self from non-self. So it seems tedious, but in that process, they're exposed to protein gene products. So your genes make proteins, and then you have all of these different proteins that are made, and these newly formed B and T cells will then be exposed to these proteins that are in your body. So it kind of tests them. It says, okay, what do you think of this one? And if the B cell or the T cell bind to it, then it dies. That's it, because it's not allowed to bind to self proteins. So only the B and T cells that don't bind to anything during this testing period or this training period, if they don't bind to any self proteins, then they're allowed to move on into the lymph. Okay, so B cells are called B cells because this training process, this self from non-self training process, happens in the bone marrow, so it's B for bone. T cells are called T cells because this training process for them happens in the thymus. Okay, so that's where that T comes from. So how does this happen? How do we have receptors, or in this case, this is an antibody? How do these things be so specific? And they have this area up here. This is the variable region. Okay, so as these receptors, there are receptors for T cells. This just happens to be an antibody, which are associated more with B cells. But it's the same process. They have this variable region that can change, and it will just change its amino acid sequence just randomly, and then or sort of randomly, and then it will just kind of try different things. Okay, and so it then pushes this out and says, okay, this is the amino acid sequence we're using. This is the shape that it is. This is, its binding ability, whatever. Pushes it out, and then if it doesn't bind to any of the self proteins, then it's just out there. It's just like fishing. It just says, okay, this is my shape. This is the shape that I am. And then if a pathogen moves in and happens to bind to it, then we're going to see what happens. Then it's preserved and is cloned. Okay. So we're going to use other terms like antigens and epitopes in addition to antibodies. So after that training, the adaptive immune system cells, B and T cells, respond to and target non-self antigens. So an antigen just is something, usually a protein, almost exclusively a protein, that is made by, well, anything. So if the immune system responds to it, then it's an antigen. Okay, so that means that if a virus starts producing proteins or a bacteria starts producing proteins, then your body's going to see those and look at those, and that's going to be the antigen. Okay, so it's anything that induces an immune response. So almost all antigens are proteins or peptides. They may recognize other things like nucleic acids, but they're not as effective at stimulating that response. So when we look at an antigen over here, we can see the antigen is this full peptide or protein, but there's only a small part of it that the immune system will actually see. So in this case, this is an antibody again. Could be a T cell receptor. But this region that it actually recognizes is called an epitope. Okay. So it would be like, you know what Paris looks like. Paris could be considered like the antigen, but you recognize it because of the Eiffel Tower. Okay, so that's the small part of it that actually makes it recognizable. Yes, the rest of it is there too, but we know what it is because of this epitope. And I think there's an Eiffel Tower in Las Vegas, so you can kind of also see how autoimmune diseases can happen and the immune system can get confused, but we'll talk about that maybe later. All right, so that's the epitope. Epitope is a specific part of the antigen that the immune system recognizes. There may be many epitopes on a single antigen, so it could be, so the immune system could recognize many of them and form lots of antibodies against lots of different parts of it. That would be polyclonal. Or it may just decide that there is only one epitope, or in the lab you may decide that there's only one epitope that it's going to bind to, and we can call that monoclonal. Okay. All right, so the presence of foreign antigen activates lymphocytes to combat an infection. Okay. So what happens when a pathogen or other foreign organism enters the body? This might look familiar. So the innate immune cells, such as macrophages and dendritic cells, are usually the first to notice. So we can see here, really it could be like a mast cell, but they get that signal, that first response. So neutrophils come in, but we're kind of interested in this macrophage. This macrophage came in early. Okay. Now, it may have been told by other cells to get there, but it came in early, and it starts to eat the pathogens or whatever's out there. Okay. So then that macrophage we're going to see is going to present that on its cell surface. It's going to be in the, we're going to talk about that in the next slide. And it's going to move through the lymph system. That's what this picture is trying to show. This is the lymph system. So it's going to move through the lymph vessels and then kind of settle at the lymph nodes. Okay, so let's talk about those antigen-presenting cells and how that works. Obviously, the antigen-presenting cells present antigens. They do that on the surface of the membrane. So this is, it's kind of faded here, but this is a cell here. Kind of something like that. And on the surface of that cell is a protein called major histocompatibility. Okay, so in the case of antigen presenting cells, this is going to be major histocompatibility, or MHC class 2. Okay, so we call these macrophages, dendritic cells, and also B cells, we call these professional antigen presenting cells. Okay, so that's their job, or one of their big jobs. Okay, so they're going to find proteins or the pathogen or whatever. They're going to break it down, okay, degrade it, and then they're going to take these protein fragments and they're going to move them up and express them on the outside of the cell. It would be like, I don't know, not to be too gruesome, but like, you know, taking your enemy and then putting pieces of it out and wearing them, and somebody saying, oh, that's Dave's hand, I recognize that. Okay, so that's what the T cells are going to do. They're going to say, you probably shouldn't have hurt Dave. Anyway, so real quick, major histocompatibility stands, or MHC, major histocompatibility complex. we're going to talk about this again, but MHC1 is on all nucleated cells, and MHC2, which is what we're going to talk about first. I know we're going in the wrong order, or in a different order, but MHC2 is present on these antigen-presenting cells. So MHC gets its name from the fact that they are largely responsible for transplant rejection. So these things were noticed to be that if somebody had a very, very different sequence to their MHC proteins, that if two different people had very different sequences, that they would probably not be a good donor-recipient match. Okay, so they didn't match genetically. So that's one of the things we look at when looking for a good match for someone in a for organ donation, is we look at these histocompatibilities. So histo meaning tissue, and compatible means compatibility. Yeah. So the name kind of makes sense. Okay. All right. So these antigen-presenting cells we just mentioned are so this macrophage, it just ate a protein, or it ate a pathogen, it broke it down, and it's expressing protein fragments, and it's going to move to the lymph system. So they digest the pathogen, present parts of it on their MHC2 molecule, and these antigen-presenting cells then continue to the lymph system where lymphocytes are waiting. Now, the MHC class 2, this is important. The MHC class 2 will be recognized by the certain specific T cell CD4, which are also called helper T cells. Okay, so the way to remember that is that 4 times 2 equals 8. So MHC2 goes with CD4. Okay, we're going to see later, kind of jumping ahead here, but it matters, that MHC1 is associated with CD8. CD8, and so that also equals 8. So I call it the rule of 8. If you can't remember whether your CD4s are associated with MHC2 or 1, you just remember that 2 times 4 equals 8, and 8 is going to be associated with MHC1, so 8 times 1 equals 8. Okay, it can help. All right, so back to this. We have our macrophage that is expressing a protein fragment on its MHC2 protein. Okay, the CD4 in the lymph node sees that and binds to it. If a CD4, after it's been trained, binds to that protein fragment, that means it is not supposed to be in the body. Okay, so it recognizes it then as non-self, and so that CD4 suddenly is important. So it has a receptor out there that's just kind of been wandering around waiting for something like this to happen, and then it binds to it, and so now it knows that there's a pathogen out there and that it responds to that pathogen. So it's going to clone itself, which means it's going to start dividing with that specific receptor, and it's going to also send out signals to others. Okay, and I have this note down here. Remember, MHC2 are always associated with CD4 helper T cells. All right, so activation. So CD4 helper T cell activation by these antigen-presenting cells. So now we have this situation where the macrophage has pulled something up to the lymph nodes, and the CD4 helper T cell recognized it as being non-self, okay, because it's trained to do that. So then it's going to clone itself, and it's going to increase in numbers. It's also going to release cytokines to further stimulate other immune cells like B cells and CD8 cells, T cells. So we can see in this picture, let's see, if we look at this one first, it's just, kind of what we expect to see. There's the receptor, okay? This is the training. This thing is not going to bind with anything self, but it does click with whatever that antigen-presenting cell brought up. So that's non-self. When that happens, what we're seeing here is the same thing. It's recognizing that little peptide. It's calling it an epitope because it's responding. And then that helper T cell is going to release cytokines and start signaling that, hey, this is a problem. We've found something here. And it's going to clone itself. Okay, so now that receptor there are going to be lots of these with that specific receptor all over the place. So if other macrophages bring them up, then then that's also going to activate it. So now we know that there's a pathogenic organism inside the body, and we're having a huge response. We're cloning that particular CD4 cell, helper cell. We're gonna activate something called the CD8 cytotoxic T cells. We're gonna activate B cells. Lots of cell division. So I have this note down here. That's where we start to see swelling lymph nodes. Okay, so lymph nodes are swelling. That's what we have down here in the corner. That's what we're saying down here in the corner. And it makes sense because that's where all of this is taking place. A signal has happened, and so we have lots and lots of cell division. Okay, so now what? Now what? We have this CD4 cell, this helper T cell that's activated, but so what? What's it going to do? Well, it's going to send out signals turning up the immune system. That's one of the reasons it's called a helper T cell. It already destroyed the pathogen, or the macrophage already destroyed that particular pathogen. So the goal is to find out where those pathogens are and to launch an offense against them. So if we recall, so what we're doing now is we're gonna start using the B cells. Okay, so we're gonna use a different type of cell to really take care of business here. So recall earlier I mentioned that B cells can also act as antigen presenting cells. That means they can do the same thing that macrophages do. They can engulf or phagocytose pathogens and then present them on also an MHC2. Now, why would they do that? Because if you know, B cells can form antibodies, okay? In fact, their receptor even looks like an antibody. We can see that up here. Okay, but it's not an antibody yet. It's just a receptor. But if it happens to recognize something that doesn't look right, then, because remember, they've been trained too. So if it binds to it, that means it's probably foreign. So then it presents it on its MHC2, okay? It doesn't just start making antibodies against it. Presents it on this MHC2 and checks in with the helper T cell, okay? That's an important thing that it does because it's like an extra check. It's like, hey, this doesn't look right to me. I don't want to admit it, but I can make a mistake. And so it checks with the CD4 cell, and the CD4 cells, yeah, it doesn't look right to me either. We're good to go. Let's kill this thing. I don't know. So it will check with that CD4 cell just like the CD4 T helper cells did with macrophages. Only in this case, when it sees it on a B cell, it activates that B cell. Okay, so the B cells phagocytose that non-self invaders, express pieces of them on the MHC2, just like they did, just like macrophages did. But, and I'm reading these bullets here now, because the B cell response is so powerful, they do have to check with that CD4 helper T cell. It also recognizes it as non-self. And so then after that CD4 helper T cell agrees that it's non-self, the B cell with that specific receptor will turn into something called a plasma cell, and it'll release those receptors as antibodies. So let's look at what that looks like. So we have the B cell that said, hey, this doesn't look right. What do you think, T cell? And so that CD4 helper T cell says, yeah, I don't think it looks right either. Activates that B cell. And so the B cell in this scenario down here, okay, that B cell differentiates into a plasma cell. And this just shows one, but it's going to start cranking out these what were B cell receptors, but now we're going to call them antibodies. And it starts just letting them go into the blood. They're immunoglobulins. They're proteins. They just move out into the fluid and they go everywhere. They go into the tissue, the blood, everywhere. Okay. Except inside cells. They don't go inside cells. We'll have to have a different mechanism for that. Okay, so plasma cells are modified B cells that produce and release copious antibodies into the blood and into the extracellular fluid in the tissues all over the body. And that overwhelming response will kill the pathogen. There's really no place it can hide except inside the cell, and then you've got other T cells that are going to get those. Okay, so any free-floating pathogen is probably not going to be around much longer. So some activated B cells will become memory cells, which will remain in the body in case that pathogen returns, because now we know that it's out there. So remember, we were randomly just making sequences saying maybe we'll find something, maybe we'll find something. Now we know there's something in the world that will bind to this particular B cell receptor. And so we just make a whole bunch of those and kind of leave them in the body in case that pathogen returns. Okay, so that means the next exposure to that pathogen is going to cause a really fast response because all those other steps didn't have to happen. Okay, that B cell or whatever is going to notice it, and then it's going to be activated just right away again. Okay. All right, so that's what this is kind of saying just very quickly. The primary immune response, that first exposure, generally takes about 10 to 17 days to occur. So that's like if you get exposed to a virus or something like that, it's about 2 weeks until your body can fight it off. So symptoms of illness occur during those days. Antigen-selected B and T cells proliferate, differentiate, all the stuff we just went through. But that second response, or all other exposures, subsequent responses, they only take like 2 days to 7 days to occur. Okay, so you may not even know that you're sick. It may just go in, and then they're like, okay, yeah, we've seen this before, and then they just start pumping out antibodies right away. Okay, and that happens because of those memory cells. Now, I will say when you are vaccinations produce that primary response. So you get vaccinated because you haven't been exposed yet, and it will produce these B cells or these plasma cells, these memory B cells. It'll produce those, so if it ever shows up again, it's there. So that's the whole idea, is to kind of put your body through that, your immune system through that, without actually having a pathogenic organism that makes you sick. So you'll feel sick sometimes because the activation of the immune response can tend to make you feel sick, but it's not doing the tissue damage that the pathogen presumably would have done. Okay, so there's our question. What happens if the pathogen just hides in the cell? I mean, what if a really smart virus says, you know what, I'm just not going to float around, I'm going to hide. Well, okay, we have something for that. So all nucleated cells, this is another thing you have to remember, I mentioned it before, all nucleated cells express something called MHC1. So this is on all of them. Remember that MHC2 is only on those antigen-presenting cells, but MHC1 is on all of the cells, including the antigen-presenting cells. So they have both. And the reason for this is that, if you remember, MHC is supposed to like display pathogens or antigens. And so that's what the MHC1 does. So all cells continuously, in this case, cleave the protein. So remember earlier, let's just look at this picture really quick. Remember earlier the macrophage had engulfed something, engulfed an organism, and then it chewed it up down here, and then it presented it on its MHC2. In this case, there is no engulfing of anything. What's going on is that you have continuous sampling of proteins that this organism is making. Okay, that's supposed to be DNA, it's making proteins. Okay, and so there are lots of proteins inside the cell, and what happens is that this kind of routinely samples these proteins, cleaves them, and then expresses part of them out on the outside wall. So it would be like if you had to display something from inside your house just randomly out on the lawn. So you put out there, a coffee cup, and then you put a lamp, and then you put, I don't know, your sofa. Just different things. You put them out on your front lawn, and then the CD8 cells, which is what we're going to see, are kind of the ones looking at this, are going to go by and say, that's fine, that's fine, that's fine. And then you put, I don't know, like an Uzi or some kind of, a cannon or something out on your front lawn. They're going to be like, okay, this might be a problem. There might be something going on in that house. So that's what it's doing. And as long as it's just putting up fragments of normal cell proteins, what we call self, then there's no trouble. Okay, but it's only if you have a virus or something and it starts making viral proteins and those get put up there, then that's a problem. Okay, because now it knows that you've been infected. So these proteins, or these T cells called CD8 cytotoxic T cells, are going to just kill it. Okay, so that's what we're going to see here. So kind of the same thing. So all nucleated cells express that MHC1. They're constantly cleaving those intracellular proteins to sample what's being made in the cells. Those fragments are then expressed on the MHC class 1 up here. And it presents these cytosolic proteins as a test to make sure the cell is working properly. Okay? Infected cells, so this is where things get different. Infected cells will have an abnormal profile of these peptides that are presented, these antigens here. Those are going to be abnormal. Okay? So, MHC1 is detected by something called CD8, and these are cytotoxic, toxic T cells. All right? So we have to remember that. Remember that rule of 8. MHC1 times 8 is 8. Okay? So that's how we know. MHC1 are associated with CD8 cytotoxic T cells. Okay? And simply put, if the cytotoxic T cell, remember it's been trained, if it says, nope, that's not supposed to be here, it will just destroy that cell. Okay? And that's it. It doesn't do anything fancy. It just kills it. Okay, so if a pathogen is hiding inside a cell, this continuous sampling of the cell content will eventually reveal the infection to cytotoxic T cells. The other fancy stuff that it does is if it does recognize something, it will again clone itself, and it will be going through, and they are, so these cytotoxic T cells do circulate, they do move around the body into the tissues, and they're going to be checking to see if they can find more of these cells that are expressing non-self or pathogenic proteins. Okay. All right, so there we go. So we're going to kind of go through all of that again. So MHC1 molecules are associated with CD8 cytotoxic T cells. So recall that all T cells are trained, so they have receptors that won't bind self. Infected cells will have that abnormal profile of peptides because it's sampling the inside, and then there's a pathogen in there, and it just happens to have sampled those proteins for that virus or whatever. So any nucleated cell that's expressing non-self antigen on its MHC will be destroyed by those CD8 cytotoxic T cells. Okay, that cytotoxic T cell, like we just said again, will then clone itself and circulate around the body. It will also, some of them will also form memory T cells, again, to prepare for a future invasion. Now they know that that pathogen exists, so it's ready for it the next time. And so that's what this image down here is showing. So it's showing the CD8 recognizing that MHC1. This is just a picture showing how it can poke holes in it. So perforins will poke holes in that cell, that infected cell, and then that cell will just fall apart. Okay, so it's destroying it. Okay, and it's more of an apoptotic type of destruction, I think. So. All right. Okay, yeah, and this is just, again, just like with the CD4s and the MHC2, it's just showing that recognition. All right, so the next question is, what if the cell just says, okay, this MHC is really, or the pathogen says this MHC stuff is really kind of a problem. It told on us with the macrophages, told the helper T cells, the CD4 cells. And now, if we try to go inside the cell, it tells the CD8 cells with the MHC1. So why don't we just make it stop making MHC? Because that's the tattletale, right? So that's what this question is. What if the cell just doesn't express the MHC at all? Okay, is it safe from the immune system then? And the answer is no. It is very unsafe. Okay, because that is a defunct or that's a cell that has a problem. So stressed cells, tumor cells, or infected cells may have reduced or absent MHC1 markers on their cell surface. Okay? So we have these natural killer cells, love that name. Natural killer cells recognize these defunct cells and destroy them. Okay? So we can kind of see over here in this picture here, we see that there's the activating receptor. Okay, it says, hey, what are you? I'm a natural killer cell. You're a normal cell, right? And then it says, yeah, I'm a normal cell. MHC1, everything's intact. That's fine. And then the natural killer cell will leave it alone. However, if it goes down here and says, hey, yo, I noticed that you don't have an MHC. Do you want to talk about that? No, I'm just kidding. The natural killer cell would just kill it. It doesn't talk. It's that tough and natural of a killer. All right, so But that's what happens if it doesn't have that MHC, then it just it wipes it out. So that's a lot of times, or many times a day, what's protecting us from forming tumors, cancers, and that kind of thing. Okay, so. And we mentioned this before, even though natural killer cells are part of that lymphocytic cell line, they're not adaptive. There was nothing special about this. It's just like, you have an MHC cell? Nope, okay. Or an MHC molecule? Nope, then you're dead. So that's not very specific. It's not recognizing specific pathogens. It's just saying that you're kind of messed up. So we keep it in the category of the innate immune response, okay, as opposed to the adaptive. All right, so other functions of antibodies that we can kind of go through quickly. Opsonization, so they can trigger these for phagocytosis. If you have antibodies that are floating around and they find something, in this case, let's see, in this case, it's just kind of covering it. Let's see, activates complement, inactive. But it will tag it. Yeah, there we go. So it's just tagging, these antibodies are just tagging it as being not okay, and then that'll give a quicker response for these macrophages or the dendritic cells or the phagocytes to eat it. Okay, so antibodies can also trigger an allergic response. We're going to come back and talk about this more, but remember allergies. Allergies are things in the environment that your body has, for some reason, your immune system has for some reason decided are not good things. Okay, and so it's your immune system that will recognize. I'm just going to say pollen in general, pollen, or it could be like a peanut allergy, something like that. But it says, okay, that's not supposed to be here. And so what it does is it will cover mast cells. Mast cells are a type of cell that kind of tends to hang out in like mucous membranes near the surface, that kind of thing. But it kind of hangs out there. And so if it has these antibodies on it, these are called IgE antibodies. We'll talk about that. But then if whatever that pathogen or whatever that foreign substance is binds to it, then it's going to cause mass degranulation of the mast cell. And so that has things like histamine, heparin. It's also got some proteases, stuff like that, that's going to trigger an immune response. Okay, and that's kind of an allergy. Also, it can activate complement. Antibodies can activate complement. We've discussed that and how it forms that membrane attack complex and can kind of poke holes in. So antibodies can trigger that. Clumping and inactivation of bacterial toxins, so it can cause them all. That's what I think we see here, is it can cause them all to kind of stick together, and then the bacteria can't do what it's supposed to do anyway, and that will eventually also be eaten. Okay, so let's look at these antibodies. I said that IgE was associated with mast cells. There are a few different types. We've got 5 types here. IgA, IgD, IgE, IgG, and IgM. Ig stands for immunoglobulin. It's just a type of protein, and that's found in your blood. So when we talk about blood proteins, immunoglobulin is one of the blood proteins. But those are your immune cell or your immune proteins. Okay. Most of the immune proteins are this one right here. This is IgG, so it's the Y-shaped one, although they're all really kind of associated with Y-shaped. But the immunoglobulin G, IgGs, are the most common, so they're the primary antibodies involved in an immune response to viruses or to vaccines. It can cross the placental barrier, so if a mother is pregnant and the fetus is able to get some of that immune response from the mother, we call that passive immunity. But yeah, you can kind of see that they all have the Y shape in general as kind of their background. But we can see, like in the case of IgM, it's a pentamer. It's a whole bunch of these together. The IgA, which is secretory, so this is what's found in breast milk, is kind of tough, kind of held together a little more, can survive going through the digestive system. So the baby drinking milk from the mother can get some of this immunity because of this IgA. Okay. All right. So this is a list talking about what these do. So IgM circulates in body fluids. This is what is going to cause a response against different blood types. This is why we need, if you're type A blood, you can't have type B or whatever. Or if you're type O, you can't have any of them, because this is the IgM is the antibody that will attack antigens on the surface of blood. Okay, so it causes agglutination or the clumping of blood. So this doesn't cross the placenta, so that's why mom won't attack the fetus, the blood of the fetus, because this IgM won't move through the fetus. Okay, IgG we just mentioned circulates in body fluids, binding antigens, so it's the most abundant. It does cross that placenta, which kind of makes a problem for a negative, because this one will attack that Rh group, and so if mom is negative and is not seen the Rh group of the fetus before, then she may start to make antibodies if she gets exposed to it, especially like at birth. If she gets exposed to that Rh factor, her body is going to start making antibodies against that Rh factor. So if she tries to have another baby that is also a positive, like O positive, that Rh factor, that Rh group, then she's going to have antibodies, and they're IgG, so they can cross that placenta and they can actually kill the baby. All right. IgA found in secretions, we mentioned that, found in breast milk. IgD, surface of B cells, so that's what B cell receptors are before they become IgG. IgE, those are the ones, I have the IgG and the IgE found on in red here, because we're going to talk about them more. But those are found on mast cells and tissues, and that can start inflammation, and those are involved in those allergies.